what is personalised medicine and the process of research. So I'll go through that. Obviously that's something that's really relevant to people. So I just, as one does, I just Googled, what do they say about personalised medicine? And so basically it distills down to healthcare on the basis of a person's unique genetic makeup. And so there's a lot of other terms in the literature and the popular press that sort of essentially mean the same thing. So you probably hear things like individualised medicine, precision medicine, genomic medicine. What they're all trying to do is get at what is a person's unique genetic makeup, because we are all different, we all look different, we're different in every gene and protein in our body. There are elements of difference between us. And what it's trying to get at is what is it about the person's unique genetic code that we can tap into that can make their healthcare better. Throughout all of our body, there are many, many cells, so they're little bits. There's 6 billion of them that make us up the way we are. And within those cells then are the things called chromosomes, and it's on those that there is the DNA, so you hear about DNA in the popular press, and it's the DNA that makes up the genes. The problem is that the genes and our genetic code actually has about 3 billion base pairs. So if you're into computers, then that's basically 3 gigabytes of data. And each data point is important. So each of those little data points, each of those 3 billion base pairs, if you just get a change in one of those, it can make a profound difference to the body's makeup or the way we behave, all sorts of factors. So this is why precision medicine is a great goal, but it's actually a very difficult thing, because we've all got variations in these 3 billion data points, and we are a combination of all of those variations. So actually drilling down to these precision aspects is a very challenging thing. So this is what we try and do from a research point of view to get at this precision medicine type of scenario. So what we're trying to do is work out for people who've got a particular condition, what are the particular features of that condition, and you know, certainly from an eye disease point of view, there's various investigations that happen. So the electrophysiology, the looking in the back of the eye, the ultrasound, so on and so forth, to try and understand how that condition is actually affecting the person. And then what we aim to do is try and get to the genetic code. Now that has been a very, very difficult thing, because as I said, there's 3 gigabytes of data and 3 billion base pairs, and each one of them is important. So up until very recently, we've had no technology to actually be able to assess that in a meaningful manner. Very recently, there's been next generation sequencing, genomic strategies, where we're able to finally have some way of accessing those 3 billion base pairs, but even that is quite difficult, because there's only about 2% of those probably that we actually fully understand. And even that we don't fully understand. There's a lot of bits in between that we still don't understand. So it's still a fairly major jigsaw puzzle to try and understand all of the genetic factors that contribute to a person's makeup or their particular condition, to try and get at this precision medicine business. Because what we want to do then is, if there's a particular condition, we want to be able to diagnose it, to provide a genetic diagnosis, to give people answers about how this happened and so on. But then we also want to get on to understanding how it happened and what we could do to treat it to make it better, if that may be possible. So these are the sorts of conditions that we work on. There's many others, but these are particular genetic eye conditions. They're not always genetic causes, but they often are. So things that like anterior segment dysgenesis and glaucoma, congenital cataracts, retinitis pigmentosa, and so on. So what is the impact of personalised medicine? So if we can get at these genetic answers, then we can help people understand. So sometimes people say, but I'm the only person in the family affected. How could this possibly be genetic? And it can be, because there can be a gene change that has happened just in that person. Because whenever any of us have children, we have to copy out our whole genetic code, so 3 billion base pairs. And so when that happens, there can be mistakes or changes that happen in that process. So sometimes a person can be the first person affected with a genetic condition in their family. Or on other times, both parents can be a carrier for a particular condition. And they're only a carrier, they're not actually manifesting the condition, so they never knew, they never ever knew. And then they can have an affected child. So there's various different types of inheritance patterns, and so the precision medicine terminology aims to try and get at that, to try and give people more information about how these things may have happened. And then, of course, we're very interested, and the research is very interested in, if possible, and it's not always possible, but if possible, working towards some therapies or treatments that might help the situation for a person with a vision problem. And of course, this is very difficult, but nonetheless, it's something that we're interested in working on. The first level is trying to find what the genetic answer is, if it is a genetic condition. And even though we've made this wonderful progress that I've talked about in terms of the genome, the way we make a genetic diagnosis and so on, and the genomic studies, from our recent work that we're doing, some of it's published, some of it's unpublished, we're still doing these research studies, what we've found in both the developmental eye diseases, in that includes the anterior segment disorders, the congenital cataracts, the microphthalmia, anophthalmia group of conditions, and also the retinal dystrophy. So that includes things like retinitis pigmentosa, cone dystrophy, Leber congenital amaurosis, various other conditions in there. And for each of those conditions, there's over 100 or 200 known disease genes. So this is why it's an enormous exercise to try and investigate these. But what we're finding is that there's, in those conditions, depending on the particular one that it may be, looking at the known disease genes, there's about a 30 to 70% detection rate. So what that means is, if it happens to be in the group of conditions where there's a 70% detection rate, that means if you take 100 people and you run this sort of genomic study, you'll find an answer in about 70 of them. So and for those families and patients where you are able to find an answer, then that can provide useful information for the family. But there is 30 where we don't. Using the current technology and the current state of knowledge, we don't know the answers as yet. And we can run every known current test and we still can't find the answer. But that's where the research comes in. We need to keep doing research to try and find the answers, because what that means that there's probably a novel disease gene that's not known yet, or a novel mechanism, some way the genome's behaving in some funny way that's causing the problem that we don't understand currently. There is still a research process, and it's a tricky process. And the problem is that once one gets into the research process, it's difficult to give a time frame, because we don't know what new technologies are around the corner that might really help us in that process or not. So it's a tricky place for people to be, because there's hope, because there's research happening, but it's really difficult to give a time frame in that circumstance. And then the other element of the research process is when we try and get onto treatments and therapies then. And again, that is an enormous process in terms of understanding how the genes work, there's an enormous number of metabolic processes and the way the genes work to make the structures. I mean, our bodies, as we know, are incredibly complex. And so the things that have to happen on a cellular mechanism and for the eye to work the way it does, it's truly extraordinary. So again, that's an enormous research process to go in through all of those steps. And I'm not obviously going to go into all of those steps today, but I think it's useful to understand the complexities that happen here. Not to say that great things don't happen, and they do, but there is an enormous research process to go through for this sort of work, which of course the SaveSite Institute is very engaged in and very positively working towards. And this is just an example of some of the steps that we take. So here I'm talking about some of the things that we do. So we might take for DNA testing, we usually take a blood sample. For some of the, to understand the function of the genes and the proteins and how they work, we often take a skin biopsy, do some sort of, sometimes we can turn those cells into stem cells, which can then mimic the eye, and we can work on drug therapies and so on from that point of view. But again, quite a long and involved process. So that's the information that I was wanting to get across about how there is this personalised medicine, and we can get very useful answers from this for families, but there is still research that's needed, lots of it, both for understanding the genetic cause and also then working towards the treatments. And I'll just put up there, that's the Genetic Services website there. There are genetic services throughout New South Wales, and you can go onto that website, and you're very welcome to make inquiries with the genetic counsellors and clinical geneticists at the various services if you would like further information. So thank you. It's been a pleasure to be here.